Okay, so this is clearly a different day. I'm in a different location. I'm wearing a different shirt. So we're now ready for Chapter 2 Notes, Part 2 of 3. So this one just kind of goes over prokaryotic cells, which your brain should say, okay, prokaryotic means bacteria. So prokaryotic cells, if we understand a little bit about them, it makes learning about eukaryotic cells a little bit easier. Um, so we'll just jump right into it. So prokaryotic cells, regardless of what type of bacteria they are, they tend to look very similar. They fall in kind of three broad categories based on shape. So you can have rod-shaped bacteria, and these are called bacillus bacteria. So that's kind of, if you think in your brain, what does a bacteria look like? It looks like a little rod-shaped cell. Okay. There can be spheroidal bacteria, so those are the cocci bacteria, so like Streptococcus, Staphylococcus. That's saying, the bacteria themselves are little spheres. Okay. Or bacteria can be spiral shaped, so there's spirilla bacteria. Um, so they can kind of come in those three different flavors. But in general, they're much smaller than eukaryotic cells. They often are able to move independently because they're just a single celled organism. They have to be able to find food or whatever all by themselves. Another cell's not going to bring it to them. Um, they tend to have cell walls, which most eukaryotic organisms do not have, except for like plants. But they do also have a plasma membrane. They have a circular DNA molecule that's not located in a nucleus, and we'll talk a little bit more about that. And a big point is that prokaryotic cells do not contain membrane-bound organelles. So, first of all, kind of the first thing that sets them apart, and that's very important, is the cell wall of a prokaryotic cell. So, when we think of cells, at least I do, I think of a eukaryotic cell, and you have here's the cell, and it's surrounded by a plasma membrane, a phospholipid bilayer. For a prokaryotic cell, they have another support system around that, and that is the cell wall. Okay, so it's very rigid. It's a rigid structure. It's not as fluid as a plasma membrane. The cell walls of prokaryotic cells, so of bacteria, are made up of a carbohydrate polymer called peptidoglycan. Okay? And there are kind of two broad ways that these cell walls are formed. And it is extremely important to differentiate between, okay, do these bacteria form the cell wall this way? Do they form it this way? So there is something called a Gram stain. And it is literally taking dye and staining the bacteria. And based on how the bacteria looks, after that process, we can kind of tell what its cell wall looks like, okay? So if the dye that's used, I think it's violet something or other, if that is used and the cell wall or the bacterial cell kind of holds on to that dye, we say that that bacteria is gram positive. Okay? If the cell, once you wash off the dye and it easily washes off that dye, we say that that cell is gram negative. So those are two huge, broad categories in the world of bacteria. So is this bacteria gram positive? Is it gram negative? Again, this is huge in the antibiotics, infectious disease realm. Somebody comes to the hospital, they have a bacterial infection. The first thing that everybody's going to look at is this a gram positive or is it a gram negative bacteria? Because that's going to determine what antibiotics will work on it or not. Okay? So why do different cells get stained different ways? What is it about them that makes them so different? Okay, so gram-positive bacteria, they kind of have the classic, okay, they have the cell membrane, and then outside of that they have a really thick peptidoglycan cell wall. Okay? Embedded in this cell wall are lots of different things that give it an overall negative charge, so the dye really sticks to that very well. So that's why it's not easy to wash that dye off. So that's why these cells are gram positive. Gram negative cell walls are a little bit more complicated. So for gram negative cells, they have, okay, their plasma membrane, and then they have outside of that a little thin peptidoglycan cell wall. And then outside of that, they have another membrane called the outer membrane. Okay, so gram-positive, membrane, cell wall. And gram-negative, membrane, thin cell wall, outer membrane. Okay, so that outer membrane doesn't kind of hold the dye very well, so that's why they become gram-negative. Okay? Whereas the inner membrane, the plasma membrane, tends to be made of, phospholipids like every other membrane. That outer membrane tends to be made up of a different kind of lipid called lipopolysaccharides. So you'll just see it abbreviated LPS. This is important because when or if that cell membrane, that outer membrane starts to break down, part of those LPSs is something called lipid A, and lipid A is toxic. So somebody is taking an antibiotic, it starts to kill that cell starts to release lipid A as it dies, and so that can make somebody very sick as well. So it's kind of important to choose an antibiotic. Okay, is this antibiotic going to be able to bind to a nice peptidoglycan cell wall and get in, or is this antibiotic going to have to go through an LPS barrier first to get in? And then when the cell dies, are they going to be sick afterwards, or with that lipid A toxin being released, or are they going to be fine? So whether or not a bacteria is gram-positive or gram-negative kind of goes a long way in the medical world. Okay, just some more definition stuff. Okay, so we have the plasma membrane, we have the peptidoglycan wall, we have the outer membrane in gram-negative bacteria. The space in between the outer membrane and the inner plasma membrane, which contains the cell wall, that's called the periplasmic space. So different stuff happens for those kind of bacteria in that space. Okay. Lastly, in terms of coatings, certain bacteria, I think both gram-positive and gram-negative, they can secrete a protein carbohydrate substance, and they use that to kind of coat their outsides, whether that's coating the outer membrane or that's coating the cell wall. The substance is known as glycocalyx, so basically a sugar shell around them. Okay? You may see it called a capsule as well. Okay? If a bacterial cell has coated itself in this capsule, it's very hard for our immune system to penetrate through that. It's very hard for an antibiotic to penetrate through that. So it's not great if somebody has bacteria in their body that have formed these capsules. When a lot of those bacteria that are forming the glycocalyx capsule kind of come together, they kind of coat like their whole family with it. That's called a biofilm. So again, very hard for our medications or our immune system to kind of break through that. So if you think of something like an abscess, abscess is where bacteria have kind of walled themselves off, and it's hard for antibiotics to get in. So a lot of times, a surgeon actually has to go in and drain that abscess and actually take it out of a person's body before the antibiotics can actually get in. Okay, so glycocalyx, biofilm, those are things that bacterial cells add to themselves to make themselves more resistant to damage from antibiotics or from our immune system. Okay? Just briefly about the plasma membrane itself. Okay, so we said that the outer membrane is made of LPS and has lipid A. The plasma membrane, or that inner membrane, or the only membrane for a gram-positive, is just like a eukaryotic membrane. It's got made up of phospholipids, a phospholipid bilayer. It's got proteins embedded or attached peripherally to it. It's just like we talked about in the last set of notes. Okay. Inside the cell, so inside whatever walls and membranes it's got going on, is the cytoplasm. I'm sure everybody's heard that term before. Okay? Whereas the cytoplasm of a eukaryotic cell is pretty busy. There's a lot of stuff, a lot of organelles, a nucleus. It's pretty full up. The interior of the cytoplasm of a bacterial cell, a prokaryotic cell, is a little more empty. There are areas where, okay, this is kind of the area where this happens, and this is kind of the area where this happens, but it's not necessarily an organelle that does it and an organelle that does it. They just kind of separate where things happen. one area like that is called the nucleoid, okay? And that is kind of its almost nucleus region. Okay? So in eukaryotic cells, our DNA is shaped like chromosomes, and it's in the nucleus. Okay? Um, in bacterial cells, the DNA is just a circle. Okay? So again, it's DNA, so it's that double helix, but it helix all the way around and forms like a nice little circle. And it's just kind of out there in the cytoplasm. It's in that nucleoid region because that's where all the stuff that needs to work on the DNA for replication or whatever, that's where all that stuff is located. But it's not really protected in any kind of nucleus or underneath any kind of other membrane. Okay? So that's kind of the main DNA, main genetic material of a bacterial cell. Bacterial cells also may have one or several, or none, plasmids kind of throughout their cytoplasm. Now, plasmids are little bits of other DNA that the bacteria may have run across and picked up. Or, its parents may have picked up and then passed it on to it. So plasmids are tiny bits of DNA that the bacteria keeps around because it gives them some kind of advantage. So that plasmid may be a bit of DNA that codes for something that helps that biofilm form and makes it harder to kill. Okay, so bacteria keeps that plasmid around. It uses, the same machinery, ribosomes, things that read the DNA, everything that it does for its main DNA, it does the same thing for the plasmid as well. It might be a plasmid that codes for something that hides a receptor that our immune system kind of keys on, so our immune system won't recognize it. Plasmids tend to be things that allow it to survive. So we're constantly worried about antibiotics not working on bacteria. So one way that antibiotics stop working on bacteria is when bacteria start to pick up plasmids to become resistant to the bacteria. Okay. Lastly, for prokaryotic cells, I did say that they kind of have to move independently. So there are things called flagella or pili or cilia, little hair-like structures that allow these single-cell organisms to move. Let's see, the book actually called them external appendages that allow prokaryotic cells to move. Wonderful. Pili are kind of like little hairs. Sometimes cells, the pili of one bacterial cell and the pili of the next one, they can kind of meet and they exchange genetic material. That's a process called conjugation. Cilia, other little hair projections that kind of sweep, food into the bacteria or sweep bad things away. And then the flagellum, there's usually just one of it. It's the big long tail structure that kind of pushes the cell along where it's wanting to swim. Okay. The only eukaryotic cell that I can think of right now that has a flagella are sperm cells. I may be mistaken, but if you can kind of picture, okay, that's what that looks like. All right. So, said all that to say, okay, that's prokaryotic cells. So they're not as complicated. The next section of our notes about eukaryotic cells, a little bit more complicated. Hopefully, I've pared it down to the bare essentials. All right. See you in a few.